Welcome to today's Monash Partners Comprehensive Cancer Consortium Precision Oncology Seminar. My name is Mark Shackleton. I'm the clinical lead of the MPCCC Precision Oncology Program, and in my spare time, also the Director of Oncology at Alfred Health. I'd like to first acknowledge the traditional owners of the lands from which we join together today, and respectfully acknowledge their elders past, present and emerging, as well as any members of the Aboriginal and Torres Strait Islander community that are gathered amongst us here today. So the MPCCC Precision Oncology Program is a community of scientific researchers, health professionals and academics across Victoria that have a shared interest in precision medicine, and particularly in delivering such an approach to patient care. The program is currently working to establish better access to molecular sequencing for cancer patients, particularly across southeast Victoria. And we also, as part of this, hold several tumour stream aligned molecular tumour boards to support the interpretation and clinical application of sequencing data derived from patient cancers. The program's been very successful with very active and consistent engagement across the research and particularly clinical communities, and we look forward to its ongoing development. Along that line, I've actually got some great news to share with you today, and that's that the MPCCC has recently been awarded funding for the next 4 years. This means that this precision oncology program will be able to continue and actually really expand in the coming months as we roll out, hopefully, at least additional molecular tumour boards and work particularly towards integrating the outcomes of the sequencing analysis processes into making more therapies available for patients. And our idea is that we'll be really expanding to include more sequencing platforms to support the research and also increase our coordination of clinical outreach across the region that we serve. So these seminars that you've attended here today are an important part of the Precision Oncology Program, and they help to share the latest research activity across the partnership, particularly in Victoria, but also nationally and internationally, and to communicate new ideas and approaches to clinical care. Consistent with that, I'm really pleased today to welcome Professor Melissa Southey, a molecular geneticist with qualifications in pathology, medicine and law, of all things. Melissa wears many hats these days. She's the chair of precision medicine at the School of Clinical Sciences at Monash Health. She's the director of Monash University's new Biobanking Victoria initiative, and also, in fact, the research director of the MPCCC itself. She's best known for her work with familial cancers and in the identification of heritable risk factors. She's certainly made major contributions to the evidence-based practice guidelines for the management of people who are at high risk of cancers, and has co-authored more than a staggering 550 papers. She leads a multidisciplinary team in diagnostic as well as research settings in Australia as well as internationally. She's got funding from many sources, from the European Commission, NIH, NHMRC, and the NBCF, and she actively participates on a number of national and international advisory, governance and editorial bodies. Most topically, her remarkable career was acknowledged in the 2021 Queen's Birthday Honours Roll with an OAM for her services to epidemiology and to precision medicine. So there's really no more appropriate speaker to talk to us today. So thanks very much, Melissa, and I'll hand over to you. There will be some questions, some time for questions at the end of the presentation, and we look forward to that. Thanks, Melissa.
Thanks, Mark, very much. I hope I'm now actually off mute and you can see my slides. And I hope now they're in appropriate view. Excellent. Okay. So thanks very much. And I'm really pleased to have the invitation to speak today and speak to this topic, which I hope will be relevant across the landscape of many of your contexts. Oops, as long as I can change my slides. Okay, so just to introduce this a little bit in very sort of simplistic, high level sense. Actually, I'm getting a little bit of information from my Anna. Is that really stable? I've got a message about unstable. We're okay.
It looks okay to me, Melissa. I can see
okay.
you slide on the screen.
Okay, I'll just wing it.
I'll let you know if anything
okay.
untoward happens.
Thanks. So here is how people, at least historically, have thought about public health overall. It's usually involves a large number of people and has historically involved a small amount of data relating to the very large number of people. It's been a discipline really that's focused on health of populations and been thinking about protecting and improving the health of people and their communities. And it's been achieved through prevention practices and interventions involving the whole of the population. So in contrast to that is precision medicine. And I think for today's conversation, when I say precision medicine, and I'll try not to say it, what I'm really talking about is precision oncology. I think we can slip between the two fairly easily. And this has been really more about a small number of people or a relatively smaller number of people and probably a much larger amount of data relating to those people to deliver precision oncology. And it's been a bit of a different pitch to public health. It's been really more about a holistic understanding of an individual's health and about creating more precise treatments for that individual, more precise prevention programs for that individual, about really targeting specific traits and profiles of individuals in a population. But as I'm sure you all appreciate, this is really beginning to change, and this is what I'd really like to talk to and put some of our work in prevention into context. So I know many of you are coming from diverse areas of precision oncology, and this, what is precision medicine is, still talked about a lot, and the literature still has great conversations about how we actually define this. And I think one of the nice things is that it is very broad, and you can almost roll anything into this discipline if you'd like to, and I think you should. So it's certainly not just about genomics. It's about lots and lots of areas of measurement, including imaging and including social aspects of our lives that can come together to deliver precision oncology and precision medicine. I've adapted this from a paper, actually not a very recent paper, but I really think for some time people have been thinking about precision medicine as a really deep and diverse collection of data that is brought together to inform medical practice. I would say that I actually have a fundamentally significant problem with this diagram, and maybe it's because it's historic, but I really do need to change this. I think we're getting better than drawing images like that, and I think we need to appreciate that precision medicine needs to deliver value to a really diverse group of humans, and we need to start changing the way that we illustrate this. But maybe that's my my tact, not yours, but I'm going to keep trying to change these images as I present. So, lots of people, as I say, have had comments about this. I really like John Maddox's comment, again, from a few years ago. Precision medicine is changing from an art of crisis management to the science of good health. And some colleagues from the US, if successful, more care will occur before disease is apparent. This shift from disease treatment to disease prevention and early detection. I think a lot of our language in precision medicine and precision public health is about maintaining health rather than responding to disease. And I think a lot of people are actually beginning to step into the realm of realizing this rather than just having it as an ambitious goal in their work. So, precision oncology and public health have certainly come together to produce this perhaps new, but perhaps not new, depending on your perspective, discipline of precision public health, which brings together the value of the really large population studies and the value of really large data sets together for the reasons really, I think, to promote individual precision medicine, but I will come back to that. So precision public health in a very high level, it's about preventing disease, it's about promoting health, it's about reducing health disparities in populations, and it's about applying emerging methods and technologies to do that. So some of the key features of this discipline really is the emergence of our capacity to both generate and analyse the so-called big data, not just in omics as I've described it across all of our omic capacities, but also more and more our studies are involving person and patient generated health data. So data collected from smartphones and watches in all sorts of ways that we perhaps didn't even imagine 10 years ago, particularly in epidemiology, but also bringing in electronic medical records and perhaps, to use precision public health speak, electronic health records into the scene is a phenomenal new capacity that is really waiting to be realised, but lots of challenges ahead to be able to do that. So it's the amount of data that we've been able to generate, but also our analytical capacity. So we're able to manage these sorts of data sets, store these sorts of data sets, share these sort of data sets overall, developing capacities in artificial intelligence, in machine learning, and in statistical and modelling capacities in disease-specific contexts and in disease contexts. Have really advanced to enable real activities in precision public health, at least as far as my perspective shows. So this really is a convergence, and I think it's given us capacity to walk around all of our questions in this space more easily and fit them into the context in which they're best placed. But I'd really like to argue to the authors who wrote this fairly recently that I think in actual fact precision public health feeds back really important information back into precision medicine and also back into public health. So to really simplify this, I think the key areas where big data is improving precision public health probably lie in these four key areas. And these four include disease surveillance and signal detection, risk prediction, disease risk prediction, targeted treatments and interventions, and general study of diseases, etiology, risk factors, biology. I think most of what we're doing fall into these four very broad categories. But in the end, at least in a clinical context, and particularly in our experience in clinical genetics, we have to bring this all back to a person. And this is still really quite a challenging thing in some contexts. These studies are done in enormous populations with enormous data sets, but in the end, we really need to deliver something back to an individual. And how we do that and how we interpret that and how we will achieve that, I think is probably our main challenge probably in the next decade, if not the next 5 years, to be able to do this precisely for an individual rather to make general comments about populations and subgroups within our population. So I think there's that challenge, individualizing the information, but I think there's also the challenge and another sort of diversity across the landscape of what we're doing is that we have to think about it across the life course. So this is a really nice figure that I've also stolen from somebody else's work that I think has thought very carefully about the life course. I mean, it thinks about the life course from preconception through to death. It's put a few fundamental milestones across the life course, which I think is a really important way of thinking about precision medicine and precision public health. So I'm just going to slip to make the conversation perhaps a bit more specific. I would like to step into breast cancer as an example of some of the elements I'd like to bring through in this discussion. And you can see here that the authors of this paper have put cancer as sort of an average diagnosis, I think, on this landscape, which is a reasonable thing to do, of course, in the context of a very broad speaking figure. But really, when we're talking about breast cancer, I think we need to be thinking about this in a much more diverse way across the landscape. And although perhaps there's an average age of diagnosis, we really do need to be thinking about managing breast cancer probably from preconception, given some of the risk factors that we're beginning to learn about in the epigenetic space. But across the landscape, so there's great opportunities for prevention pre-diagnosis. And some of the risk stratification is probably best based in earlier parts of life or teenage parts of life. I think that's a conversation that will generate more data in the coming years, but we need to think about this across the life course. And for those that are diagnosed, of course, the later parts of life about disease management, risk management, monitoring of disease and so forth becomes very important. And we are beginning to develop lots of tools across this landscape to actually deliver breast cancer risk management across the life course. So to come back to these 4 elements for breast cancer, I think there's some really nice examples in all 4 of these areas for breast cancer. In the context of disease surveillance and signal detection, I think mammographic density, working mammographic density has really demonstrated that imaging can provide very important information for risk estimation for women. In risk prediction, which I'll come back to, many of the genetic factors are now being used in a combined risk model, which has been very helpful to place women more precisely on the risk spectrum. Certainly some of the germline information and somatic genetic information is informing targeted treatment options for women diagnosed with breast cancer. And there's so many examples of how precision oncology approaches has given us more insights into breast cancer, the heterogeneity of the tumours, the heterogeneity of the disease, the subtype specific issues, and so on and so forth. So I think it's a nice example to speak across these 4 elements of precision public health. But for today, I think I would just try and, again, bring the conversation in a little bit to give you an example, talk to risk prediction. And to think about risk prediction and breast cancer, I think this is a really nice way to illustrate the issues. Breast cancer, this describes breast cancer across the lifetime risk and also against relative risk. Whoops. So you can see that for the population-based sample represented in this diagram, the blue line, that against the average risk, that most women actually lie below average risk. But for the red line, which represents women at high risk of breast cancer, you can see that there's a really big tail that rolls out to the right of this diagram, which is why the average risk sits where it is. But for those women, many of those women have considerable risk of developing breast cancer during their lifetime. And to bring that into a figure that talks about what we know about genetic risk factors for breast cancer at the moment, here's another illustration which has taken some liberties, but I think is a useful way of presenting this in this context. So thinking about relative risk versus the variant frequency, there are some genes like BRCA1 and TP53 that we know have very rare pathogenic variants. But when women carry very rare pathogenic variants in these genes, they are at considerably increased risk of developing breast cancer. And there are other genes that sit further down this illustration, BRCA2, PALB2, and further down again, ATM and CHEK2, that we know again have very rare variants in the genes that are associated with perhaps, dare I say, more moderate risk of breast cancer. And there are all these exceptions to the rules, I suppose, coming through. So we do know about some variants in BRCA1 that appear to be associated with less risk of breast cancer than the average BRCA1 mutation does. And a really extreme example in BRCA2, that is a protein truncating variant, but associated with considerably reduced risk compared to the average risk of the BRCA2 variant that sits up in this area of the diagram. But an important part of this that has really been generated via genome-wide association studies that have needed to be internationally coordinated to be able to have the power to detect these variants are these very common genetic variants that are associated with very small increments of risk, which sit down in this part of the diagram. And there's a very large number of them. At the moment, most models are based on 313, and we could have a very long conversation about how many of these there are, but there are at least 313 that provide really important information about risk associated with breast cancer due to what they call a polygenic risk score. So for the, oh, I'm sorry, that slide's going backwards. Let's go this way. So when we add all of these risk factors together, it only explains about 50% of the familial risk of breast cancer. And for the women that carry the most of these very common genetic variants, these women sit about here on this diagram. They're at about a 3-fold increased risk of developing breast cancer. So these are the really the women that have the largest number of these common genetic variants that together come together to increase their risk about 3-fold. And interestingly, this risk is associated, they're likely actually more likely to develop ER positive disease than ER negative disease. But look at this again in a slightly different way. This rectangle represents early onset breast cancer in women with a very strong family history. And for these women, this is actually real data generated from the Australian population. We know that a proportion of these women carry pathogenic variants in BRCA1, a proportion carry pathogenic variants in BRCA2, a small proportion of these women carry pathogenic variants in other breast cancer susceptibility genes, including PALB2, ATM, CHEK2, PTEN, for instance. But still, the majority of these women were unable to identify a pathogenic variant in a breast cancer predisposition gene and to so-called explain the breast cancer, their personal breast cancer and the breast cancer in their families. But we are, of course, building up a really good picture about the complexity of breast cancer predisposition. And we know that there are several elements beyond a rare pathogenic variant in a breast cancer predisposition gene that come together to make up a woman's risk of breast cancer. So we know, as I've said, that this includes common genetic variants associated with breast cancer risk, the so-called polygenic risk, and this is relevant to every woman in this rectangle. I've told you very briefly about mammographic density. It's a very strong breast cancer risk factor and it's relevant to every woman in this rectangle. And there's emerging evidence about epigenetic risk factors, which again are relevant to every woman in this rectangle. So for a woman that might sit here, that has a pathogenic variant in BRCA1, she might have a very large number of these common genetic variants that come together to make a polygenic risk. She might have very high mammographic density. We're beginning to understand that this woman is probably at the extreme end of the risk for breast cancer. And for a woman who sits here that might have a pathogenic variant in PALB2, have a collection of the polygenic risk variants, moderate mammographic density, she might sit more in this part of the risk spectrum with considerable uncertainty about exactly where. And then a woman down on the right-hand side of this rectangle, for which we don't know very much, we might be able to measure her polygenic risk, and we might know a bit about her mammographic density, but not knowing anything about any rare variants in a breast cancer susceptibility gene. It's actually very hard to place her on the risk spectrum at all. So I guess since the late 1990s and probably for the next at least 15 years, the transition of this information into clinical genetics has been fairly methodical. There's been the classical sort of accumulation of evidence and the translation into practice. This has been supported by Everq Cancer Treatments Online, which has been an online reference for best practice guidelines for clinicians in this space, so that at least around this country, women with the same genetic risk factors are told standardised and expert panel agreed information about her risk. And there's been a continuous improvement as we've learnt more about these genes and the risk associated with carrying variants in these genes and the best risk managements and so forth. The risk of second primaries, for instance, this information's been used to update best practice guidelines. So that was fairly methodical, but in the last 5 years or so at least, gene panel sequencing has really disrupted this process. And we very quickly reached a point where gene panel sequencing became very popular. Some of it was delivered to the population directly. It was obviously the technology provided it at a cheaper and quicker rate. In the 1990s, I remember a BRCA1 test used to take us 6 months in the laboratory, literally, to be able to sequence the whole of the BRCA1 gene. Now it could be turned around probably in a matter of days or weeks. With a number of other genes on these gene panels using massively parallel sequencing methodology. So it's the testing is no longer needing to be limited to a small number of genes, and that it caused this sort of almost explosion of genes being put on panels and women being tested for all sorts of genes, some of which we didn't know very much about their variation. We certainly didn't know anything about whether their variation was associated with susceptibility to breast cancer. So with that in the background, we and others really needed to catch the evidence up with the practice rather than the other way around. And the Australian-based initiative to this was called BRASTRAP. As you can see, it involved a very large number of collaborators and contributors, which have been linked to most of the clinical genetic services in Australia and linked to international activities being conducted in the US and throughout Europe with very similar underlying designs, and was really pitched at being able to understand the genetic variation in genes that were being included in these panels. So this has been funded in Australia by the Breast Cancer Foundation. It's been funded in Europe via the European Union Horizon 2020 program, and in Australia, luckily via an NHMRC European Union Collaborative Research Grant awarded to Mandy Spurtle and myself to support these initiatives. So just to give you a little bit of information about BRASTRAP, it includes about 30,000 Australian women. Those women have come really from 2 different sources, firstly from genetic services. So these actually include the women who've ever stepped forward to these services for genetic testing for breast cancer predisposition. So this has occurred in Australia since the late 1990s. These women have consented to testing, their DNA has undergone testing and their DNA has been stored. We've been really fortunate to be able to access that DNA, and some of it has had very historical testing. So, some of the testing that was done in the 1990s was only the protein truncation test and only looked for protein truncating mutations in some parts of the BRCA1 and 2 genes. So some of it was very historically limited. And we've been able to go back to all of these DNAs and apply a gene panel test. So those are the clinical genetic services collaborating with BRASTRAP. And the other engagement we've had has been with large research resources in Australia. So the Australian Breast Cancer Family Registry, the Melbourne Collaborative Cohort Study, ASPRI, and the Variants in Practice initiative, which has really been a collaboration between the clinical genetic services in Victoria led by Paul James. So we set out to consolidate these DNAs in the biorepository. In the team, this was an extraordinarily laborious process, which we achieved fairly quickly, actually, thanks to a lot of hands-on and enthusiasm to get this done. This DNA was we applied a gene panel test that included 24 genes, the genes you can see there in pink. We did this via a couple of different technologies for reasons I can go into if you're interested at the end. Most of this work has been led by Tu, Ewan Dumont, very ably within the team. So having done this sort of scale of testing, we immediately had in our hands information that was returnable to the clinics and returnable to the women whose DNA was in the study. And this was done back to the clinical services, and the information's been made available to the women via those services via routine clinical genetics in the centres that were collaborating. So that was sort of partly because of historical testing, but also because we had additional genes on the panel by then, which some of these women had never been tested for. But the main thrust of BRASTRAP was, of course, to be able to interpret more information from the genes we knew less about, and that's really been done via international collaboration. Predominantly with Bridges, which is the EU-based activity in this space, and also with Enigma, which is a consortium focused on finding ways to better classify rare variants in breast cancer predisposition genes. And the mission of all of this, of course, was to provide more information, more precise information about more variants in the genes we knew about, but also about genes we didn't know much about, via FEQ back to the clinic, so they could manage the information being delivered by these panel tests. So at this point, I'd just like to limit the conversation to one gene, which has a bit of an interesting story, I think, and that's PALB2, which, you know, really should have been called BRCA3, I think, but it's PALB2. It was first described as a breast cancer predisposition gene by Naz Rahman and her team in the UK, having done a smallish case control study within a cases selected with a strong family history in 2007. It was already known as a protein that interacted with BRCA2. For that reason, it was a candidate, and it was conducted in the context of what I guess we used to call candidate gene analyses. But it was really given a very unfortunate tag, and that was a tag of it being a moderate risk gene, which is really the story that I'm going to embark on. So with that tag, and it was really from her early work that gave it that tag, PALB2, there wasn't a lot of attention really put to it, even though it was a really big discovery, at least from many people's perspectives. I think it just had this overtune of not being perhaps so important. But it soon became evident, and we were part of actually bringing it to light that, you know, at least for some variants, the risk associated with some rare variants in PALB2 was really, the risk was really considerable. And our first analysis only involved a few families within the Australian Breast Cancer Family Study, and we had found a few families carried rare pathogenic variants, and this, and actually in all families that we found in our early work, they all carried the same variant, and it was this 3113G to A. So with James Doughty and others, we were able to estimate penetrance using information about first and second degree relatives, and using MISC models. So using polygenic background plus PALB2 as a major gene, we were able to estimate penetrance just using really 5 families that carried this variant. So the estimate of penetrance to age 50 was 49% and to 70 was 91%. Please note the very large confidence intervals. But really this was the first indication that rare pathogenic variants in PALB2 were not associated with what we sort of were thinking about as a moderate risk of breast cancer. We also did some work with colleagues in Finland who had found a mutation in PALB2, who had a population-based collection of families that carried this founder mutation, and with them we estimated penetrance to age 70 of 40%. And just to put you into some sort of picture of perspective, at the average penetrance of the BRCA2 pathogenic variant to age 70 is 45%. So we were really looking at a gene where pathogenic variants were much more like BRCA2 than I think we realised when PALB2 was first described as a breast cancer predisposition gene. Since then, there's been really large international efforts to try and consolidate and understand the breast cancer risk associated with pathogenic variants in PALB2. This is one of the attempts. Just as we were doing the work that I described to you about estimating penetrance, we were designing ICOGS, which was a Illumina custom array for genotyping in a large international consortium. And we were able to argue to put some rare variants on these arrays, which was not really done at the time, and it did take a little bit of conversation, but we were really interested in measuring some of these rare variants frequency in the population to get a better handle on the risk associated with carrying them. So you can see the sort of international engagement we were engaged with to be able to measure this on this scale. And here's just the results from this paper looking at the PALB2. This is the 3113, the W1038X variant. It was genotyped in a very large number of cases and controls, and the odds ratio was nearly 6. The variant below is the Finnish founder mutation, again, very similar data. So another piece of evidence that these variants weren't associated with moderate risk, they're really associated with much more considerable risk. So after this work, we were able to, in the Australian context, have best practice guidelines available on Evicure around genetic testing and risk management related specifically to this variant. And again, it was a little unfortunate the way it rolled out. It wasn't that we were thinking this variant was the only variant associated with considerable risk, but it was the variant we'd found in the Australian families, and we had very little information about other pathogenic variants at the time. So we were able to take a step forward with best practice guidelines really limited to this one variant. But it was clear we had to do more work, and we really had to do it in another international context, and thus the PALB2 interest group was born, which, as you can see, gained really considerable international interest and willingness to collaborate to get to the bottom of the risk associated with carrying pathogenic variants in this gene. So there are actually 2 significant papers published by this group now. This is the second one. This involved 524 families from 44 study centres across 21 countries. So some of them came from multiple case family studies and some of them came from population-based studies. It accumulated a very large data set of which to do this analysis, as you can see. In this analysis, we restricted the variants to pathogenic protein truncating variants. And of the 161 families in this study, 61 actually carried this variant that we had identified to be identified in the Australian families, this C3113G.A. And we excluded all missense mutations. So it was a fairly classical modified segregation analysis adjusted for ascertainment and looking for the best genetic fit model for using a similar methods to what I described before for the work that James Doughty led. So this is the findings. So for breast cancer, this really large study found to age 80, a 53% penetrance of pathogenic variants, protein truncating pathogenic variants in PALB2. There was some evidence for a cohort effect. We also found evidence, and this was the first time that we really found definitive evidence for there to be a risk of ovarian cancer and a risk of pancreatic cancer associated with carrying pathogenic variants in PALB2. The ovarian cancer risk and the pancreatic cancer risk is considerably lower, but it is statistically significant, and of course very important to know given how difficult it is to manage pancreatic and ovarian cancer. So this is a table from the paper, and sorry for the complexity, but the most parsimonious model included a residual familial component for breast cancer and ovarian cancer, which is illustrated really in this table. So if we just look at the bottom, so this is cancer risk to age 80. So breast cancer risk to age 80, you can see from without considering family history, the risk is 53%, which is what I showed you in the previous chart. But you can see when you consider families with different family histories, so this, for instance, 71% to age 80 for a woman who has a mother affected at age 35 years or below. You can see that family history has a considerable impact to the penetrance estimate. So family history still has a really big role to play in the clinical management of these women, because there is a background familial component still to these risk estimates. But having demonstrated much more definitively that the risk was not limited to that one variant that we'd found in the Australian families, but to all pathogenic protein truncating variants, we then were able to work with EvoQ and change the guidelines to include information for genetic testing and risk management for carriers of all pathogenic protein truncating pathogenic variants in PALB2, which was a really great step forward. So for this woman in the middle that carried a PALB2 mutation, this work did allow us to estimate her risk a little more precisely. It did push her up to the right-hand side somewhat, and it still did range from around between 50 to 80%, dependent on family history, as I've just shown you in that chart, but we were more, it was more definitive that the risk was associated with more variants than just the one variant that was involved in our early studies. So that does beg the question about missense variants, and I probably don't have a lot of time to go into that, but there was really a lot of early evidence from work looking at PALB2 in a number of populations that there wasn't evidence for missense variants being associated with risk of breast cancer. But with data from the very large international activities that are now underway, there is emerging evidence that at least some missense variants in PALB2 are associated with increased risk of breast cancer. But the important information that's needed to really individualize this information is still emerging. You know, which of these variants are associated risk is still a question. The magnitude of that risk is still a question. There's no evidence to suggest that the risk will be any different, but we really haven't got to the point of demonstrating that yet. There's an enormous activity in the context of functional studies to try and characterize how missense variants are disrupting the functions that we know are associated with the PALB2 protein. And there's enormous activities, again, internationally coordinated to incorporate this new functional evidence into ACMG variant classification rules. This is predominantly being led by a variant curation expert panel based in the ClinGen system, but there are other interesting activities going on outside the scope of ClinGen as well, in terms of interpreting this data to be able to individualized clinical classifications for missense variants in PALB2. So just to show you the scope of this challenge, here's just a couple, really just a couple, small proportion of the missense variants that have been described in PALB2. There's been a cascade of different biological functional tests applied to them, which are across the top of this table. In some people's hands, in some studies, there's been demonstrated functional impact for these missense variants, in some there hasn't. There's subtle differences about some of the methodology that might explain this, but still these boxes represent variants for which there's really conflicting information. It's very hard to interpret at the moment, and we need to work on to validate these studies more thoroughly. But there is a very large amount of activity in this space. I've shown at the bottom here some of the studies that are reporting the outcomes of some of these functional studies, which will no doubt bring together a body of evidence that will move this activity forward. So just guess to nearly finish with, I'd just like to come back to this diagram and still thinking about PALB2 and thinking about how the polygenic risk score might modify PALB2's the risk associated with carrying a PALB2 pathogenic variant. As I've shown you, there clearly is an impact of the magnitude of risk associated with family history, and some of that may indeed be influenced by the polygenic risk. So this is a really nice figure, I think, that's come from a recent publication from Mark Robson and his team based at Memorial in New York. And he was really addressing this question. He was really interested in how the polygenic risk might modify the lifetime risk of breast cancer for pathogenic variants in a number of breast cancer predisposition genes. And I guess one of the reasons why I chose PALB2 to bring through in the conversation today is because PALB2 is such an amazing feature of this illustration. So here is PALB2, and as our work with the PALB2 interest group suggested, the average risk for the average PALB2 pathogenic protein truncating variant is probably about 50% to age 80. But what this diagram really nicely illustrates is when you put this in the context of the polygenic risk score, how this gets pushed across the landscape of risk. So women down in this part of the curve who have a very small number of these common risk variants, their risk is really considerably reduced. I mean, nearly 20%, right? Nearly down to 20% to age 80. But the women who have a very large number of these common genetic variants, their risk is pushed up towards 80% to age 80. And this is really sitting, as you can see, under what we understand as the average lifetime risk for a carryover pathogenic variant in BRCA1 and BRCA2. So, I mean, in part, this is probably bringing out the family history story, but there's probably more components to this than I've described to you today. But nonetheless, this is really striking. So without the polygenic risk score in your risk estimate for a woman, it really still is an estimate with a real amount of wobble across the landscape, as you can see here illustrated by this red curve for PALB2. So to these are really all important elements of being able to bring all of this activity across all the components of what we understand as risk factors for breast cancer back to an individual. So I think we do need to do this. Of course, we can't bring the volume of data for every individual. It's just not going to be practical. We need probably, I think, to get to the context where we have a small amount of data that is critical to personalising this information for an individual. And for breast cancer, I think we're getting close to understanding what those components are. Certainly mammographic density is one of those important components. I haven't had a lot of time to talk about it today, but it's a conversation in itself. I think rare variants in breast cancer predisposition genes are another important component. So I think I've illustrated to you. I think the polygenic risk is also another really important component. And I think family history is still an important component in many contexts. This probably brings in other aspects like lifestyle, hormone exposure and other elements that probably aren't articulated here, but I think family history still belongs in this group of really important pieces of information that come together to be able to estimate for an individual what their risk of breast cancer is. And indeed, this is really what the most recent and now online risk prediction tool is doing. It's bringing together information, as I've described, to generate a risk estimate, individualized risk estimate for women. This is available online, and it has experienced considerable clinical uptake, particularly in Europe, where it was developed actually with the support of the European Union that's within the program that it did all the genetic testing via a panel test that I described earlier. But there is still obviously a wealth of challenges in this space. And I think three key challenges are these. I think although the aspiration and the intention, and I think eventually we will realise in precision public health to reduce disparities in populations, I think at the moment probably we've enlarged them. A lot of the key information that I've described on the right-hand side is not available for populations outside of those of European descent. We're beginning to generate it, I think, in other populations, but we need more time to generate the equivalent information to be able to make that sort of information available to people of non-European descent. I think there's real challenges, and again, there's activity in this space already, and I think we'll get there, about the implementation of the polygenic risk score into the clinical setting. There are a number of active trials in this space that are beginning to learn and understand how this might be best done, but I think there's a lot of work still to do and trials to finish to be able to achieve this. And there's ongoing and maybe forever ongoing challenges around classification of genetic variants that are characterized in activities such as gene panel tests that really do need more systematic attention, because I think we're moving from a situation where empirical evidence is going to provide sufficient evidence to make them clinically actionable. I think and into activities like functional studies that need to be so well validated and internationally standardized to be able to incorporate them into best practice classification for clinical use. So with that, I would like to acknowledge that, of course, this work has been done by tribes of people across the globe, not just me. I hope I've represented their work appropriately today, but in the way that I have. There's been a large number of funding bodies, as you can see, really great colleagues who have done critical parts of this that has made it achievable in our context for our population, and I hope promotes precision public health into the future. So thanks very much.
Thanks so much, Melissa. It's a real tour de force. We're very proud of that work and your role in it. It's great to have you part of our community. There's a few questions popping up in the chat, which we'll just swing straight to. We've got a couple of minutes. So this one's from Antonella Papa. Do you ever find more than one variant co-occurring in the same patient, such as PALB2 and P53? Many thanks.
Yes, look, not that commonly, of course, but of course you don't do testing on this scale without finding them, and they're a complete challenge. Someone
Yep.
with it, yeah.
Next question from Michael Abramson. Thanks, Melissa. What is known about gene X environment interactions in breast cancer? Is there any advice we can offer women who carry these PALB missense variations about behavioural and lifestyle modifications? So is there a role for environmental factors in increasing the predisposition to developing breast cancer?
I don't think there's...
So if you have a predisposing mutation and you're a heavy drinker, are you more likely to get breast cancer than if you're not if you're a teetotaler?
Yeah, I didn't have alcohol on my critical list. Maybe I should have. It's often one I'm asked about. Look, I think when you carry a pathogenic variant, all of those factors become more important. So all of the other risk factors become more important. There's probably not a lot of data in some aspects for me to have said that, but I think generally there's a lot of evidence generally to think that that's consistent across all risk factors.
Next one's from Puya Faridi. Thanks, Melissa. Do you think in the future we can develop precision preventive cancer vaccines specific for genetic variations? I mean, you'd need the mutations to be immunogenic, and they wouldn't be if they're in germline.
Correct. Look, I think there's in some contexts, I think there's some promise for chemoprevention. I think that's probably a space that will move forward first. It gets a bit beyond my understanding. Perhaps, I could put the question back to you. I mean, there are some disciplines, of course, and where gene therapy and even CRISPR is being considered, but it's a bit space age, I think, at the moment. But, if we can make things more precise and the off-target things less of a concern, I think, there's clearly a space for that. Some of it probably belongs in the pre-implantation space, and that's another whole conversation. But, I'd like to think that we'll be clever enough to have some really good strategies for these. But again, I don't think breast cancer, for instance, is probably at the forerunner. I think some diseases where the penetrance is higher and the consequence is higher really should take the limelight in some of these activities first.
I mean, certainly, yeah. I mean, I guess certainly there's, I think, a reasonable amount of optimism in the targeted therapy space for, you know, identifying or targeting therapies that are specific for signaling pathways that are predicted to be acted by specific germline mutations, probably a bit more so than vaccines at this stage. Another question from Vicky Marshall: what are the theories behind mammographic density as a risk factor? Why are they linked?
I think they can be put into two basic baskets. One is that the density masks tumours, and that's a bit different. But I think the part of mammographic density that's a risk factor really needs a lot more exploration. I think people are beginning to understand what the information, at least from digital mammograms, is that predicts risk. But biologically, I don't know whether someone like Cara Britt is online, or would be better to answer the biological underlying consequences. There's a lot of work in that space, and there's some really interesting mouse models where they're transplanting human breast tissue and being able to maintain the density and do really staggering stuff, but I'm probably not the best person to speak to it.
I'm getting cold sweats about my PhD, which we thought that was all about, but that's a long time ago. I've just got a quick question to finish off. So, I mean, one of the striking things is one of your first slides, one of your early slides, which shows the proportion of, you know, kind of card-carrying heritable breast cancer risk for which we have no idea what the underlying driver is. Do you think that really is going to be full of true kind of polygenic risk, or do you think there are other parts of the DNA molecule outside of coding regions that we need to be interested in? Is anyone, I mean, are there kind of large-scale whole genome sequencing studies, maybe applying, you know, AI to the analysis of those data to try and find other clues in the DNA molecule as to why some of it's a predisposed?
Yes, there's a lot of large-scale but differently focused activities, of all of which you've described, Mark. I think personally, I think the answer is going to be a combination of all of those things. I think there's a lot of rare variants that we aren't able to classify yet. I think there's a lot more information about polygenic risk. I think there's more information in heritable epigenetic risk factors, which again, I didn't get into today, but, you know, I think the answer is a number of things which would, I mean, for the last 20 years, we've sort of been chipping away at this slowly, and my rectangle that I showed is slowly filling up, but boy, it's slow, but it's exciting because every step forward, you know, has the next sort of cascading effect.
Great. All right, well, we're out of time. So thanks so much, Melissa, for your presentation. Again, it's a real privilege to have you as part of our community, and thanks so much for your work and for talking today. Thanks everyone also for attending. These seminars have actually generally been fantastically well attended, and it's great to see that momentum continuing. The next seminar will be on the 27th of August. Put it in your diaries right now. And the presentation will be from an international guest, Professor Felix Feng, a radiation oncologist from the University of California. Please keep up to date with upcoming seminars and other activities related to NPCCC via the website, which is Monash Partners, monashpartnersccc.org. And also subscribing to our newsletter and tuning into the NPCCC socials as well. Have a wonderful rest of the day, everyone, and weekend, and we'll see you at the next seminar.